Moving on from the esophagus to the stomach. Different regions of the stomach that you should be familiar with. You should pretty much know everything in this figure. It's not a lot. Esophagus, of course, which we were just looking at. And this region of the stomach is referred to as the cardia. I'm not exactly sure on the historical significance of naming it the cardia, but it is close to the heart. And if there is gastric reflux and acid moves out of the stomach back up into the esophagus, if that occurs and we have gastric reflux, the acid burns the esophagus and it feels like the heart's on fire, because the heart is right behind that. So perhaps that's the reason that this part of the stomach is referred to as the cardia. They used to be referred to by some people as the cardiac sphincter, but we're not going to talk about that. That's just confusing. It sounds like something in the heart. This is the gastroesophageal sphincter that occurs at this point. It's a fairly weak sphincter. It's relatively weak. It's formed in part by leaves of the diaphragm, sheets of muscle that are, there's a hole through the diaphragm, the esophageal hiatus that allows the esophagus to pass through from the thoracic cavity into the abdominal cavity. But the diaphragm muscle, they're like little layers, thin layers, and they help to form part of that gastroesophageal sphincter. The fundus is the upper portion of the stomach, shaded in yellow, and that can be variable. It can be rather large in some stomachs and less significant in other stomachs. And most of the time when you see these stomachs drawn, they're shown like this one is sort of an artist's conception of a normal, average stomach. But some stomachs have a very pronounced and large fundus where it moves quite a bit upward. It's quite pronounced. It's not a big deal, but there's just individual variation. And of course, the diaphragm still goes over the top of it. So all of the stomach, of course, is in the abdominal cavity. So at any rate, that's the fundus, acts sort of as a storage spot. Then there's the body of the stomach and the pyloric region and the pyloric sphincter. This is a strong sphincter. If we were dissecting fetal pigs, you would feel the pyloric sphincter, and it's a large muscular structure at the exit of the stomach, controlling exit into the small intestine. Two other things on the stomach is the greater and lesser curvature of the stomach. The greater curvature is just on the outer surface. It's larger, of course, and then the lesser curvature of the stomach, just for orientation. On this slide, it's a little bit busy, but we're looking at different cell types of the stomach mucosa. So the illustration shows a little section of the stomach wall cut and then enlarged in the diagram. So at the top of the figure is the opening down into one of these pits, and the lumen of the stomach would be here. So the inner surface, so the food, mucus, material, liquid, is up at the top of the figure. And then there are lots of these little pits, these glands, like crypts, and they descend deep into the mucosa of the stomach, and they're lined with lots of different types of cells. So there are a couple cells that we'll take a look at. There are mucous cells in sort of the neck of this duct, and they secrete mucus. Goblet cells. Okay, lots of mucus produced, and there is bicarbonate in the mucus also to help buffer the acid so it doesn't wear through the mucus and start to erode the lining of the mucosa. There are parietal cells that we'll take a look at, and there are a couple of functions that I want you to pay attention to for the parietal cells. They produce hydrochloric acid. The parietal cells. You might remember parietal has an L in it, so does hydrochloric. And they also produce intrinsic factor. Other cells that we'll take a look at are chief cells. And chief cells make the digestive enzyme pepsin, which is secreted in its inactive form as pepsinogen. So it's pepsinogen that's released, goes out into the lumen, where pepsinogen is then converted to the active protease pepsin. So at the bottom of the figure are those couple of highlights, as I've already mentioned. Parietal cells and chief cells, and then there are also several paracrines that are released. Local acting hormones, gastrin and histamine are two. And they are produced in different cells of the stomach mucosa, and I'm not asking you to remember the name of those cells that produce those two hormones, although gastrin is pretty straightforward. G for gastrin, so those are the G cells. The histamine I don't really care about, as long as you remember that it is produced in the stomach mucosa. And we'll take a look at its role shortly. So, chemical digestion of proteins begins, and as I just mentioned, parietal cells produce the HCl. And the role of the acid is to denature protein. It interrupts hydrogen bonds. It's strong enough to cleave certain covalent bonds, so it starts to unwrap and unfold the protein. Chief cells make the pepsinogen. And pepsinogen, here's that ogen ending again. And when you cleave off the part of the polypeptide, you're then left with pepsin, which is the active protease. So pepsinogen is sort of a pro-protease, if you will. The goblet cells are producing mucus and bicarbonate. Something else to note, as I mentioned on the other slide, is that parietal cells are also producing intrinsic factor. Maybe I didn't mention it on the previous slide. But intrinsic factor produced in the parietal cells goes into the lumen of the stomach and moves on into the ileum, the small intestine, and is necessary for the absorption of vitamin B12. So it's a different role. And vitamin B12 is needed for red blood cell formation. So, in elderly people, the parietal cells don't produce as much intrinsic factor as they used to. So if you have decreased intrinsic factor, that can lead to decreased vitamin B12 absorption, and that can lead to pernicious anemia, where the person just isn't producing enough red blood cells. It's fairly common. So what would be a reasonable treatment for somebody who has pernicious anemia from lack of intrinsic factor? Yeah, they just take a vitamin B12 supplement. And then that helps with the red blood cell formation. Okay. So intrinsic factor produced in the parietal cells of the stomach targets the small intestine for absorption of vitamin B12, which is necessary for erythropoiesis. Let's do a little bit on the production of hydrochloric acid by the parietal cells. So this is a parietal cell that we've just looked at, and it's producing hydrochloric acid. So some of this should look familiar. First of all, let's just start with the signals. So there are three stimuli or signals for the production of hydrochloric acid. And when all three of these are present at the same time, it sort of turns on HCl production like opening up a fire hose. So gastrin, acetylcholine, and histamine, they each have a receptor on the cell surface, and they have a cell signaling pathway, involves a protein kinase. Okay, and that's enough of that for the moment. This is just showing a cell signaling pathway. Perhaps you did some of that in ANP1, I believe you did, where you were looking at cyclic AMP, adenylate cyclase, etc. So that's the story going on there that we won't go back to right now. But follow the rest of it. So the protein kinase gets going, and then we start to produce the acid. See if you can follow along with the actual production of the acid. It starts with what we've done before, carbon dioxide and water are combined, and there's the enzyme carbonic anhydrase. This should look familiar. It produces carbonic acid, and then the carbonic acid ionizes and produces the bicarbonate ion. Let's just follow that for a moment. The bicarbonate ion that gets produced inside the cell. This should look familiar. So as the bicarbonate ion is produced, it's moved out of the cell into the extracellular fluid, and as the bicarbonate leaves the cell, the chloride enters. So there's our old chloride shift again. Okay, and then the chloride just continues on and is moved out of the cell, and there's half of our hydrochloric acid. There's the chloride ion. Let's go back to the carbonic acid that was produced by the anhydrase. The diagram should have shown the hydrogen that came off. So here is the hydrogen that came off when it ionized, the bicarbonate on the right, hydrogen on the left, and that should have gone on up like this. And this shouldn't be there. Okay? So the hydrogen comes to the luminal side of the parietal cell, and see if this looks familiar. The hydrogen gets actively pumped out. At the same time, a potassium is moved in. And it's this primary active, so there's a hydrogen potassium exchanger. That looked very similar. It's the same basic machinery that we saw in the A cells of the collecting duct when we were looking at acidosis. And so this is the proton pump. We're exchanging hydrogen for potassium. And then the potassium just exits again. We don't really worry about it. So at any rate, there is our hydrogen now in the lumen of the stomach. So it started again with the formation of carbonic acid, CO2 and water. Move the hydrogen out of the cell back into the extracellular fluid. When the stomach starts producing a lot of acid, it makes a lot of that bicarbonate and is sometimes referred to as the alkaline tide because the blood leaving the stomach is more alkaline. It gets buffered eventually, but it is a byproduct of producing the acid. Okay, I would like you to read about Helicobacter pylori, or H. pylori, and its role in peptic ulcers. So there's a little bit of that in the textbook. You shouldn't spend more than about 5 or 10 minutes reading about it, but I would like you to be familiar with Helicobacter pylori. And the last slide looks at inhibiting acid production in the stomach. So if you remember, let me back up 2 slides. So we're going to be looking at the histamine and the proton pump. Remember, there's ACH and gastrin also for other signals, but that's not what we're going to be talking about in this slide. So, H2 blockers. An H2 blocker, it's a class of drug. It acts as an antagonist and blocks the histamine receptor so that histamine can no longer bind to the receptor. And if histamine cannot bind to the H2 receptor, then the parietal cell is not stimulated as much and it produces less acid. So 2 common drugs are Zantac and Tagamet, and they act as an H2 blocker, preventing histamine from stimulating the parietal cell. There's another class of antacid medications that are the PPIs, and these are the proton pump inhibitors. And they just inhibit that hydrogen potassium exchanger, and the cells push less hydrogen into the lumen of the stomach, and they are very effective. So that's it for this section. We'll pick up with a little bit of digestive phases in the next short lecture.